In our previous session, we looked at this, the iconic double helical structure of DNA, and we tried to pick apart the forces which hold it together, and discovered really that it's a very complicated picture and that we can't focus just on, for example, the hydrogen bonds between the bases, which are perhaps the most famous part of the explanation for the structure, but that we had to think about the interactions with water especially in order to understand why this structure was stable. Now what we're going to do this time is to sort of pick up those ideas a little bit, but focus on the idea of the specificity of base pairing. So we saw that an A base always pairs with a T one and a G always pairs with a C. And we said that that was linked to both the compatibility of their hydrogen bonding groups, but also the geometry of the pair that was formed as a result, which had to pack properly into the structure. Now what we're going to do today is to ask, how is that specificity of base pairing achieved when DNA is formed, in other words, when it replicates in the cell? And we're going to look at how difficult it is to achieve the level of accuracy that's needed to sustain life. And then we're going to look as well at what the consequences can be when errors are made and the wrong sequence is copied during a DNA replication event. And we'll see what the cell can do to deal with that problem and what the consequences might be if it fails to deal with it, which includes the possibility of the development of cancers. So what we know is that within the long double helical structure of DNA, we have this specific pairing of bases. So when one strand has a T, that's always hydrogen bonded across in the other strand to an A. And when one strand has a C, that's always hydrogen bonded across to a G. And so you get these complementary DNA sequences on the two strands. And that's hugely important because that sequence encodes information which basically determines what the cell is able to do and how it does it. So part of this long sequence of base pairs is a code that determines what the sequences of amino acids will be in the proteins that make up the cell, for example, all of the enzymes that control the biochemistry of the cell. And that sequence of amino acids determines the properties of those enzymes. If there are mistakes in the DNA sequence, then some of the amino acids may be incorporated incorrectly into the protein, and that can change or indeed completely abolish the activity of those enzymes with huge consequences for the viability of the cell. So the accuracy of the DNA sequence is incredibly important. Other parts of the sequence are involved in determining how those proteins are produced in the cell, so controlling when they're produced and at what level. And again, that's hugely important for making sure that the cell works in an efficient way. So again, errors in those parts of the DNA sequence can also have quite profound consequences for the cell's properties. So the big issue happens when a cell divides, because when a cell divides to produce two new daughter cells, the DNA has to be copied so that each of the daughter cells has what we hope will be an identical copy of the DNA in the original cells. And that process is called DNA replication. And it's that problem of how to achieve fidelity in DNA replication. In other words, how to ensure that we get as far as possible, a completely faithful copy of the original sequences in the new cells that we're going to look at. And what we're going to see is that that's a formidable problem, and that biology is incredibly good at it, but not perfect, because it's just essentially impossible to achieve it perfectly. And as I say, we'll look then at what the consequences could be when it goes wrong. Now, what you don't get from a sort of simplified schematic like this picture is a feeling for just what a formidable task this is in terms of the scale of the operation. Because we're not just talking about a few hundred base pairs here. we're talking about a human genome which has 3.3 billion base pairs in it. That means that if we just take one set of our chromosomes, between them they have 3.3 billion base pairs, but of course we have two sets in a cell, so that's 6.6 billion base pairs in total have to be copied. And of course each of the individual bases within the pair has to be copied. So that's actually something like 13 billion copying events that have to be correctly carried out every time a cell divides. 13 billion is an awesome number. And it's not just the size of it, it's the fact that it has to be done quickly. The total time for replication of a genome is around an hour in a human cell. So that means to achieve 13 billion copying events in an hour, things have to move very quickly. And the individual enzymes that make this reaction happen generally process something like 50 bases per second, which is pretty fast. But of course, if you just have one enzyme trying to do the whole thing, that would take a ludicrously long time. So actually, you have multiple enzymes sort of working simultaneously and then stitching their work together at the end. It's a tremendous feat of organization. And through all of this, accuracy is the key. So that's what we're going to look at now. Now, we're not going to look in a huge amount of detail at how DNA polymerization works today, because it's a phenomenally complex machinery that does all of this, and it's something that is well worth looking at at a later time if you're interested in this, because it is an extraordinary mechanism that's been evolved for achieving this. But we just want to focus specifically on the issue of how we get specificity of base pairing here. So we're just going to take a very sort of simplified view of the process and then focus on the specificity aspects. So this protein, which is about to start the process off, is called Helicase. And its job is to grab a hold of this double-stranded DNA and pull the two strands apart. Because, of course, if we're going to make copies of the individual strands, we need to separate them from each other so that each one can act as a template for building a new complementary strand. And because the double helix is an energy minimum state, the helicase is going to need to find energy from somewhere in order to pull the two strands apart. And so the helicase uses the energy from breaking down ATP in order to separate the strands. So let's watch it do that. So there goes the helicase. It latches onto the DNA, and you can see it starts to pull the strands apart like that. And that's the first essential step in the polymerization process. Now what's going to happen now is a bunch of other proteins assemble adjacent to the helicase. We're not going to worry too much about the function of these ones for the moment, but we'll just let them do their thing. But the one that we are going to be interested in is this one. Now this is the DNA polymerase. This is the enzyme which actually catalyzes the formation of the new strand. And so what you'll see is that the DNA polymerase processes its way along here, following in the wake of the helicase, and adds new nucleotides onto an ever-growing daughter strand there. Okay, and there it goes, and you can see the new DNA being formed as the DNA polymerase moves through there just behind the helicase. Something similar will happen on the other strand, which is called the lagging strand here, though that's actually much more complicated for reasons that we won't get into here, but the chemistry of the polymerization event is actually the same in both. So let's now look at that in a bit more detail, because that's the first thing that we have to think about in terms of how do we make sure that the right base is incorporated at each position here to pair up with the right one there. So here we are then poised just about to add another nucleotide onto a growing DNA strand. So what we have here is, first of all, this is the template strand. So this is the existing DNA strand of which we're trying to make a complementary copy. This is the new strand that we've made, and right at the end of it, this is the last nucleotide that we've added. So we have the base, which forms its complementary interactions with the corresponding base on the parent strand. We have the sugar residue here, and this link goes via a phosphorus onto the new strand there. Also attached to this sugar, we have this OH group, which is very important because that's what's going to react in bringing the new nucleotide in. So let's have a look at the reaction. This is what's going to happen. So DNA OH represents this. So this is our new DNA molecule, and this is the OH there, which is going to be involved in the reaction. What it actually reacts with is this. This is a nucleoside triphosphate. So although it only needs one of these phosphate groups in order to make the connection to the existing DNA strand, it comes in with 3, and that's actually very important because what you can see happens in the reaction is that it uses one of those phosphate groups to condense onto the existing DNA molecule, and in the process the other 2 phosphate groups are released as this ion, which is called pyrophosphate, and it's the formation of that actually that makes the whole thing energetically favorable. So the reaction has to happen like this in order to be chemically feasible. Okay, so let's now bring that nucleoside triphosphate in. So there it is. So there's the base, forming, we hope, complementary interactions to the corresponding base on the parent strand, so that we'll get a faithful copying process. We've got sugar residue here, and then here are the 3 phosphate groups. And the key is that we're going to end up getting a new covalent bond across here between this oxygen and this phosphorus, and that's what's going to be necessary in order to integrate this into the molecule. Now, if we didn't have an enzyme, the chance of the correct nucleoside triphosphate, in other words with the correct complementary base here, arriving here close enough for these 2 atoms to react together and hanging around long enough for that reaction to happen, that probability is vanishingly small. So basically nothing happens in the absence of an enzyme, just for probability reasons. And that is something that enzymes are very good at dealing with. Enzymes have lots of strategies for catalyzing reactions, but perhaps the most important of all of them is that when enzymes bind molecules in their active site, so they bind them and lock them in place in the active site of the protein molecule, and when they do that, they can lock them so that the atoms that need to react are in close proximity to each other. So let's now see what happens when we do this in the context of the DNA polymerase enzyme. So this bit in blue is a part, just a small part of the enzyme, and you can see it involves a bunch of carbon and oxygen atoms from the protein molecule, but also a pair of magnesium ions which are hooked up to the protein molecule in the active site. And you can see those magnesium ions are crucial because they form a bunch of interactions with the triphosphate group. So they lock that whole triphosphate group in place, but crucially, they also lock this oxygen from the DNA strand that we're trying to extend in close proximity. And because this assembly is interacting both with this oxygen and with this phosphorus, it locks them into striking distance of one another so that the reaction can take place. So the next problem is that in order for this oxygen to attack the phosphorus, it effectively has to lose this hydrogen, because if it didn't do that, it would gain a positive charge, and that would raise its energy to a very high level and make the reaction infeasible. So somehow we need to find a way to lose H+ from here. And when the product is formed, that H+ is actually going to be joined on over here to form the pyrophosphate ion. So the key is, how can we move H plus across from here to here. And again, that's something that enzymes are very good at. So enzymes are very good at moving H plus ions around the active site. And we can see how it's going to do that here. So B here represents a basic group. It's a group which is part of the enzyme and which is capable of picking up H plus by using this electron here, pair here, to form a dative covalent bond to it. So B is a basic group that's part of the enzyme. And we can look then at how participation of that base allows the reaction to happen. So these curly red arrows here represent movement of a pair of electrons in each case. So you can see this lone pair of electrons on the base is going to be used to pick up this hydrogen here. That allows the bond between the oxygen and the hydrogen to break, and that electron pair is going to be the key one that then forms the new bond here between oxygen and phosphorus. So the participation of the base has freed up this electron pair and allowed it to form the new bond that we need. At the same time, we have to break off the pyrophosphate group, and that's what this electron pair is doing. So the PO bond breaks, and that pair of electrons moves out onto the oxygen to create a negative ion there. So the consequence of all of that is that we get this. So we have the new covalent bond. So this base is now integrated into the strand via this new covalent bond. And we formed a pyrophosphate ion here, which at the moment is still hooked up to this magnesium here. And we can then easily complete the mechanism by allowing the pyrophosphate group to escape, taking this proton with it, and restoring the base to its original form. There we are. We've transferred the H plus across to here. So the base has been restored to its original form, ready to participate in a new cycle. And the pyrophosphate has been released, so that the magnesium here again is now free to coordinate to a new triphosphate group on the next incoming substrate. So that's, in very simplified form, how the mechanism of the reaction works. But the question is, what does this have to do with specificity? Because the question we're asking is, how do we manage to make sure that we get faithful incorporation of the right base at this position? And the answer to that has to do with the fact that for this mechanism to work, everything has to be exactly in the right place. This oxygen and this phosphate have to be locked by the active site in close enough proximity that it's easy for this new covalent bond to be formed. And in order for that to take place, this base also has to be in exactly the right position so that it's able to pick up the H plus from here and allow this oxygen to make its attack. If things are slightly out of position, the rate of the reaction will plummet. And that's the crucial thing, because what happens is that when the nucleoside triphosphate binds at this position, if it's the correct one, so if it forms the correct pairing interactions here and it has the correct shape, then the way it can act with the protein around it allows the protein to lock into position so that the base is in exactly the right place, the oxygen and the phosphorus are locked into proximity. If the wrong base goes into there, then it can't form the right interactions with the protein around it. And the consequence is that the protein doesn't lock down into position properly. And so the oxygen and the phosphorus may not be close enough to react. The base may not be in the right position to pick up the proton. And so the reaction either doesn't happen at all or happens so slowly that by the time there happens to be enough energy for it to take place, the nucleoside triphosphate may just have fallen off. So the specificity comes from the fact that you have to have the correct base pairing here in order for the shape of this part of the structure to interact properly with the protein around it and allow it to lock into the active shape. And that complementarity, that shape complementarity, is actually very, very effective at ensuring the precision of incorporation of nucleotide. Experimental evidence suggests that this ability of the active site of the DNA polymerase to distinguish between the correct and the incorrect base by only bringing the catalytic groups into the correct proximity when the correct one is bound, that's an extraordinarily effective way of making sure that we minimize the number of errors in introducing the new bases into the sequence. It's reckoned that about 1 in 100,000 incorrect bases is incorporated, which make it past this first line of defense. And that's pretty impressive if you compare that with typing, for example. An excellent professional typist might expect to make maybe 1 character in 1,000 incorrectly. So we're talking about 100 times better fidelity than a good professional typist. However, what we've got to weigh that up against is that the human genome is huge. So although only 1 base in 100,000 may be copied correctly, there are an awful lot of bases to be copied. So let's just do a quick calculation to see how big a problem this is. So as it tells you here, the haploid human genome size is about 3.1 times 10 to the 9, so 3.1 billion base pairs. Now what haploid means is that if a cell has only 1 copy of each, or 1 version of each chromosome, like a gamete, it would have 3.1 times 10 to the 9 base pairs. But most cells, of course, are diploid, which means they have 2 versions of each chromosome. So you've got to take that into account. And you've also got to remember that when a base pair is copied, each of those individual bases has to be copied. So the number of reactions in which a new base is introduced is not 3.1 times 10 to the 9. You've got to factor in those additional issues there. And then each time that individual event happens, there's a probability of 1 in 10 to the 5 that the wrong one will be introduced. So just see if you can crunch those numbers together and just make a prediction of roughly how many errors you'd expect to be made in the sequence each time a cell divides. So pause the video, have a go at the calculation, and then when you come back you can have a look and see what the answer is. Okay, so there we are. So if it's a diploid cell, as we say, it has 2 times 3.1 times 10 to the 9 base pairs, and then we have to double that again because each individual base has to be copied. So we get 1.2 times 10 to the 10 bases have to be copied. So if each of those copying reactions has a probability of 10 to the minus 5 of being an error, then we can just multiply that probability by the number of events, and that gives us a scarily large number. 120,000 mistakes would be made every time a cell divided. So now let's just push that on a little bit further and see how serious that is in terms of the whole organism. So we've got this number that we've just calculated, that the number of errors per cell division is 1.2 times 10 to the 5, 120,000. The average lifetime of a cell, you can get this figure of about 7 years. That number is hugely variable. For some cells, it's much shorter than that. For a red blood cell, it's only a couple of months. For some cells, it's much longer. So that's an enormously variable number, but that's quoted commonly as a sort of average lifetime as a cell. And the total number of cells in the body is about 37 trillion, so 3.7 times 10 to the 13. So taking those 3 numbers, see if you can estimate how many errors you would expect to be made in your body every single day. So again, pause the video, see if you can calculate it and see what how your number compares with what I got. Okay, so first of all, we can convert that average cell lifetime from years into days, and so just by multiplying by 365 we get 2,500 and odd days is a sort of average lifetime of a cell. So if the number of cells is going to stay approximately constant, that means every time a cell dies, we need to have a cell division in order to create a replacement cell. So in effect, that average cell lifetime is also going to determine the rate at which cell division has to take place. So we're going to say that each cell on average is going to have to divide every 2,555 days to make up for the death of another cell. And so we can work out the probability that a cell divides on a given day as just 1 over that number, which gives us 3.9 times 10 to the minus 4 is the probability of a cell dividing on any particular day. And because we have 37 trillion cells, we can multiply that number by 37 trillion to find out how many cells are actually going to divide on a given day. And we get 14 billion, okay, which is an astonishing number. In your body, every day, 14 billion cells divide. So because you've got this replication frequency of about 1.2 times 10 to the 5, so 120,000 errors per cell division, then if you have 14 billion cell divisions, you can multiply those together and you get this scary number of 1.7 times 10 to the 15. So 1.7 million billion errors accumulating every day is what you would have. That's not what you do have, but it's what you would have if biology relied totally on discriminating the right nucleotide being processed in the active site of the DNA polymerase enzyme. That number is way too high for life to be sustainable. So what we need are other mechanisms to come in and improve that frequency. And what we haven't exploited so far is the stability of the double helix, because remember that discrimination that we've talked about so far depends purely on what's happening at that one position where the incoming base arrives and gets processed. But what biology can do is take advantage of the fact that every time a correct nucleotide is added, the newly created base pair becomes part of this stable double helical structure that forms. If an incorrect base is incorporated, it's going to disrupt that double helical structure. And so biology uses two separate mechanisms now to exploit the regularity of the double helix as a test of whether the correct nucleotide has been incorporated. And what we'll see is that those two mechanisms massively improve the fidelity, in other words, massively decrease the number of replication errors made. So we're going to have a look now briefly at those two mechanisms. So in this animation, what we're visualizing then is the DNA polymerase enzyme here, represented by the sort of brown blob. And then here is the DNA. So the blue strand is the template, so that's the existing DNA. And you can see that up to this point, we've succeeded in making a complementary strand by introducing new nucleotides and joining them together. So we're poised now to bring some more nucleotides in and hopefully base pair them correctly and extend the strand here. So we can just watch that happening. So in comes another one like that. And up to that point, you can see we've now added 3 more and they're all correct. But the question is then, what happens if an incorrect nucleotide comes in? So this is just giving you an example then, that a correct base pair would be something like AT, but the question is, what happens if a G comes in instead of an A? So here comes the G, and you can see the initial thing is what we already talked about, which is that sort of hangs around there, but it doesn't make the enzyme click properly into position. So the chance of it being successfully reacted chemically to join the chain is much lower than normally. And what will tend to happen is that it will just fall out again and hopefully be replaced by a correct one, which is what's happened there. But sometimes you get this scenario where the incorrect one does get chemically bonded. As we've seen, that happens about 1 in 100,000 times. So let's then look at what happens now that we've got the incorrect one bonded in place. And you can see that all of a sudden, something quite dramatic happens, which is that the protein senses the error and it completely expels the end bit of the newly created chain, not just the single nucleotide that you've added, but a whole short sequence here gets spat out from the active site and moves a long way across the protein down to a second active site down here. So it's part of the same protein molecule, but it's a completely separate active site. And if we look at what happens there, what we'll see is that instead of adding a nucleotide, it actually chops the wrong one off. Having done that, and actually it usually chops more than one off, but the principle is right, it reverts to the original active site and we go back on to adding new nucleotides again. So by detecting the error, shifting the wrong sequence down to the new active site, we're able to chop out the mistake, bring it back, and then continue the addition of new nucleotides, hopefully correctly. And that process is called proofreading. Okay, so it happens immediately, incredibly short time after the initial mistake has been made, because it all happens within the same protein molecule. The way the polymerase detects a mismatch and therefore initiates this correction protocol, which is the key to proofreading, is dependent on the idea that if a correct base pair is formed, it will immediately snap into place as part of the regular double helix that is just being extended as nucleotides are added to the chain. The idea is that if a wrong base is incorporated, that will distort this helix, and that has a knock-on effect further back in the chain, because if the helix is distorted at the point where the newly incorporated nucleotide is interacting, that will also affect the positions of the bases 1, 2, 3 further back in the structure. So what you have is a sensor which actually pokes chemical groups from the protein into the groove of the DNA helix, about 2 bases back from where the new one is being added. And so long as that sensing group is able to make contact with those bases in the right way within that groove, then it allows the helix to remain in place. But as soon as there's any distortion, so that those contacts between the sensing group and the DNA are broken, then it's sort of spring-loaded and the DNA is flung out of the active site and across to the proofreading site to have the mistake corrected, and then it's allowed back in to have another go. So it's exploiting the regularity of the double helix, which depends on all those factors that we explored last time. So the effect of this is that it's estimated about 99% of the errors that are initially made by the enzyme incorporating the wrong base in to the chain are corrected. 99% of them are corrected by this proofreading mechanism. So that means that we can increase our replication accuracy by a factor of 100, which is really good. Clearly, that's a spectacular improvement, but it's nowhere near good enough, because we saw that we were getting a frightening number of errors in each replication, and although that's improved it significantly, it's nowhere near good enough to to be continued on a regular basis. So there needs to be another mechanism to come into play, which will take place after the polymerase has done its job, left a mistake behind, something else has to come in and clean up the mess. So let's now look at what happens if the polymerase makes an error and introduces a wrong base and then fails to correct it by proofreading. So the polymerase is continuing to make its way through the genome. This is the new strand that it's created, or one of them, and you can see when we get to this point, we've generated a mismatch. So we've got a G here paired with a T where it should be paired with a C. And the question is, what's going to happen now? The polymerase can't do anything about it because the polymerase is now long gone. It's far away down the DNA strand. But that doesn't prevent other proteins from intervening. So what we're going to see now is a mechanism called mismatch repair, in which other proteins are primed to scan for errors like this. And here they come. And they recognize the irregularity in the double helical structure. So we get this combination of proteins here, which are able to bind to DNA when it has been bent out of shape as a result of the mismatch. And if we just continue, then you can see that once that's been detected, it does something about it. So this exonuclease binds and it just chops out this whole region, which included the mismatch, like that. And then, once we've got a clear space there, a new polymerase comes in and fills it in, this time correctly. So that mechanism can do what the DNA polymerase failed to do the first time round, which is detect the error, chop it out, and replace it with the correct sequence. That's actually not as easy to do as it might seem, because, of course, the big question is, how does the polymerase, how does the mismatch repair system know which strand is correct? Because all it knows, all you can see is that there's a mismatch. We've got a G paired with a T where it should be paired with a C, but how do you know whether we need to correct the G or whether we need to correct the T? And the answer to that in bacteria is well understood. In eukaryotes, as far as I know, it's actually not very well understood. But it seems like it has to happen very quickly before the DNA has been sort of fully processed. And as I said at the beginning, we haven't talked about the whole process of DNA assembly in polymerization because we've just focused on the immediate reaction. But the overall process tends to result in sort of bitty DNA, which has gaps in it that have to be sealed and things. And it seems likely that these proteins are able to recognize the strand which is new because it has unsealed gaps in it. But I don't think anybody really knows that. But the consequence of that sort of mechanism is that this has to happen very shortly after polymerization is complete, because otherwise the two strands become indistinguishable and you don't know which is the right one and which is the wrong one. So mismatch repair adds further improvements to the fidelity, and it's reckoned that it picks up about 99.9% of the errors which are still there after the proofreading has had a go at it. So when you combine those two processes of proofreading and mismatch repair, you get a decrease of about a factor of 100,000 in the mismatch rate, which is a huge, huge improvement. So if we can summarize where this takes us with our numbers game, remember we started with this result that said that each individual reaction in the polymerase active site has a probability of 10 to the minus 5, in other words, 1 in 100,000 of making an error. And if that was the only mechanism for getting accuracy, we saw that although that sounds quite impressive, it would lead to something like 120,000 errors in each cell division, which is completely unacceptable. So scaled up to all of the cells in the body, that gives this terrifying number of 1.7 times 10 to the power 15 errors every day accumulating around the body. But we've improved that situation dramatically by recognizing that these two mechanisms of proofreading, which is done by the polymerase itself, and then mismatch repair, which is done by other proteins which come in immediately in the wake of the polymerase, their combined effect improves the accuracy by a factor of about another 100,000. So now, instead of 120,000 errors per cell division, we're only going to get just over 1 on average per cell division. And that's a huge improvement. So that, looking at that number, that doesn't seem too bad. You know, one mistake in over 3 billion base pairs is really a sort of awesomely successful process. However, because you've got this enormous number of cells in the body, it still gives us this rather big number of 10 billion errors a day being made at different places around the body. So all the time we are accumulating DNA which has errors in its sequence. So what we need to look at now a little bit is to try and gauge how much of a problem that's likely to be. If you look at a relatively simple organism like a yeast, so S. cerevisiae, which is the organism whose chromosome, or part of whose chromosome 3 is mapped in the top part of this picture, what you find is that most of the DNA actually codes for something. So the blue blobs in the diagram there are the coding regions that actually encode the sequences of proteins, and there are also, as you can see, some other coding sequences that code for functional RNAs and things. And there are just relatively short sequences in between that don't have a specific coding function. However, one of the really big surprises when you look at the human genome is that it's really not like that at all. So the bottom part of the picture shows a similar map of part of chromosome 11. And again, the blue regions represent the coding regions, and you can see that now they're interspersed with great long sequences that don't actually code for proteins. That's going to be very important when we're thinking about the likely effects of the mutations we're talking about. Now, quite what the function of these sequences in between the coding regions is not all that well understood. It's certainly not true to say that they have no function at all. Some parts have quite well understood roles, for example, in controlling the expression of the genes. So what that means is that not all of the genes in a cell will be active at any one time. Some will be active in some tissues and some will be active in others, and some will be active all of the time, but some will only be active in response to specific signals. And all of that regulation of the gene activity is going to be controlled by mechanisms that involve these sequences in between the coding regions. And because not all of that is very well understood, it's difficult to quantify exactly how much of the total genome actually has a specific function. But people are sort of tending to say that it's somewhere in the region of 10% is likely to be important. So in effect, what that means is that 90% of the mutations that arise as a result of the errors in replication that we've been talking about, 90% of those probably have no real effect. And so that's obviously important. If we now take our approximate number of, 1 error per replication, we can now say, well, yeah, but that's only 0.1 error that's going to affect a functional region per replication. So in other words, only 1 error in every 10 replications. So that's, a major difference. So let's now think about what might happen if we do get a mutation in one of those regions that codes for a protein. So this picture shows you the sequence of a gene that codes for lysozyme, which is an enzyme that has a role in protecting against bacterial infection. It's found in tears, for example, and in mucus. And you can see along the top row in each part of the chart a sequence of bases. Remember, C, A, G, T are the 4 bases. So this is the gene sequence along the top. And the capital letters underneath represent the particular amino acids that are encoded by groups of 3 bases. So, for example, I wanted to have a look at this base here, which is, this sequence of 3 bases here, which is called a codon, GAG, and that codes for this amino acid E. Now, E is short for glutamate. So this is an amino acid which has a side chain which, under physiological conditions, is negatively charged, and it plays a crucial role, this particular glutamate, in the catalytic mechanism of lysozyme. So suppose that a mutation happened and instead of GAG, this became GTG. Well, we can look at what happens as a result of that, because the genetic code, the correlation between these nucleotide sequences and the amino acids they code for, that's very well known. So we can look at what happens if GAG changes to GTG. So here's a table that shows you the genetic code, and you can see our original GAG down here codes, as we said, for a glutamate residue, which is this negatively charged residue, which the particular one we looked at in that sequence lies right in the heart of the active site of the enzyme and plays a key role in the catalytic mechanism. Now, if that changes as a result of a replication error, if that changes to GTG, you can see that now that will code not for glutamate but for valine. And valine is very different. Instead of being negatively charged, it's a neutral, non-polar residue, so it has very different structural and functional properties. And that mutation would likely abolish the catalytic activity of the enzyme, or at least massively reduce it. So these single base changes can sometimes have a catastrophic effect on the activity of an enzyme. However, you can see that doesn't always happen, because, for example, if GAG was mutated to GAA, it would have no effect at all, because it would still encode the same amino acid. Now, here is a representation of what a lysozyme molecule looks like when it's folded up into its active structure. So it's a long polymer chain which has this very specific intricate fold, and this structure here, this is this glutamate side chain with this negatively charged carboxylate group here. This is another amino acid also crucial in the catalytic mechanism. And these two are in this region of the molecule, which is where the active site is, where the reaction happens. And you can sort of get the impression from this that large parts of the molecule are actually far away from the active site. Now, they may have all sorts of important other roles in making the structure stable, because remember, the precise positioning of these amino acids is crucial for the catalytic effect to work. So we can't say that these other amino acids don't play a role, even though they're not in the active site, because it could be that mutating them would alter the structure subtly and mean that these weren't quite in the right place. But it is also true to say that a lot of the amino acids, particularly ones not close to the active site, can actually be mutated quite a lot without having a major effect on the activity of the enzyme. So again, you can see that just because we have a mutation in the gene sequence, it won't necessarily have much or any actual biological effect. It depends on what particular nucleotide is mutated and whether that codes for something which is absolutely functionally crucial. Okay, so let's go back to playing the numbers game a little bit. And every time we've done this, the numbers have been sort of ballpark figures. But I should really emphasize that in this next bit, the ballpark has kind of expanded to the size of a small continent. So these are absolutely not reliable numbers. It's just an exercise to get you thinking about the sort of magnitude of the issues we're talking about. Okay, so we're going to focus on a liver for a change this time. So if you take an adult liver, it typically has about 1.5 times 10 to the 11, so 1500 billion cells in it. And the average lifetime of a liver cell is about 400 days, so it's a lot less than that 7 years that we took as our average, because liver cells are very reactive, and in particular they spend a lot of time dealing with toxic substances, and that limits their lifetime. So 400 days is a sort of typical lifetime. So if we're going to keep the number of liver cells constant, then we can say that after 50 years of life, typically a liver cell will have gone through maybe 45 cell divisions, to taking account of those numbers. Now, from what we worked out earlier, we said that every time a cell division happens, we get an average of around 1 error in the genome per cell. But we've seen that because the coding region of the genome is only about 1%, and even if we take account of regulatory regions associated with the genes, we could estimate that probably no more than about 5% is really crucial for regulation. The probability that we get an error in one of those parts of the chromosome is only about 0.05, and taking account of what we've just been talking about in the lysozyme example, the probability that an error in one of those regions actually has a serious effect on the way the protein works, or on how much of the protein is produced, again, is probably quite small. So I've just sort of guessed a figure of about 1% again, so 0.01. So if you put those numbers together and we multiply the one replication error that happens each time by those probabilities, we get a estimate for the number of damaging errors each time a replication happens of around 5 times 10 to the minus 4 for each genome. Now, if we then take account of the number of replications that have happened for a given cell and the number of cells in the liver, we can then multiply those together and come up with a number of about 3 billion for the number of damaging errors that have arisen in liver cells as a result of replication errors after 50 years. 3 billion is about 2% of the total number of cells in the liver. So that's saying that there's actually a huge number of errors that have accumulated which are in some way affecting the function of the liver. But it's still only 2%, so it's probably most of the time not going to be having a very noticeable effect, because the remaining 98% of cells are pretty much as they started out still. Now, as I say, there are massive margins of error on that, but it kind of makes the point that all through life we are accumulating errors which can have noticeable biological effects on individual cells. And there is some belief that that is part of what causes aging. So the accumulation of damaging mutations in cells over a period of years just means that the enzymes in those cells don't always work quite as well as they should, and the ways they interact with other cells and signal to one another don't work quite as well as they should, and this kind of thing. And that may be a reason why the body just functions less well as you get older. It's certainly not the only reason, and it's hugely contentious as to what the dominant factor is, but that theory of accumulated mutations is one of the important theories of aging. Okay, so what we want to do now is to see how all of this might relate to cancer. And in order to do that, we need to understand a little bit about what cancer is. Now, that's obviously a huge topic, and we're only going to barely scratch the surface of it today. But we can at least get some indication of how this thinking that we've been going through is relevant to it.
The important thing to understand about cell division is that cells can't just do it when they feel like it. It needs to be incredibly tightly controlled, because otherwise, if a cell just decided to keep on dividing, it could produce large numbers of cells in a place where they're not needed, and they could start, damaging the surrounding tissues, potentially blocking access to things, and indeed using up all the resources by outcompeting other parts of the body, and that is what happens in cancer. So cancer is a disease where these control mechanisms, which make sure that cells only divide when there's a need for it, when those mechanisms break down. So when you're growing up, obviously the cells have to divide more frequently in order to facilitate growth, but in the adult phase, what would normally happen is that cell division is basically just a maintenance process. So as some cells die off because they've sort of reached the end of their normal lifespan, they will become replaced. And there are all sorts of mechanisms for sensing when that happens, sensing the need for a replacement, and therefore switching on cell division in a local area just until the problem is dealt with. So the way it can go wrong is sort of illustrated schematically in this picture here. So this is just an example. This is how a colon cancer might develop. So it starts with the normal epithelium. So the epithelium is the layer of cells that sort of lines the gut. And as I say, normally it has this very regular, well-maintained structure. And then you get mutations starting to accumulate in cells, or starting with one cell in the epithelium. And you can see in the early stages, what that does is just leads to a sort of local distortion in the structure of the epithelium. But then as other mutations get added, the problem becomes worse, and the control over the cell division gets more damaged. And so you get more serious distortions until eventually you start to lose control altogether, and then you can get the cells dividing in an almost uncontrolled way and producing a very large mass, which is what a tumour is. And then in the sort of very late stages of cancer, cells can break away from the tumour and establish themselves in other parts of the body and produce secondary tumours. That's the metastasis phase. And at that point, the cancer is very, very serious indeed. Now remember that when we took the liver as an example, we estimated that the number of functional mutations, in other words, mutations that have potentially a significant effect on cell function in liver cells after 50 years, might be something like 3 billion. Now, of course, most of those won't have any effect on cell division, because most of those genes that are affected have completely different roles, which in no way impact on triggering the division of cells or anything else linked to it. So we can sort of modify that number to take account of that. The total number of genes coding for proteins in the human genome is around 20,000. And from studies of the mutations that occur in cancer patients, we can estimate that in a typical cancer, there's probably only around 10 of those genes which are commonly mutated and which seem therefore to be important drivers of the development of a cancer. So if we just multiply our 3 times 10 to the 9 mutations by the fraction of genes which could be implicated in generating a cancer, we come up with a number of 1.5 million. So that looks like a very scary number, because again, that suggests there are 1.5 million cells there in the liver which seem to be poised about to lead to the development of a tumour, and obviously that can't be right. And the key to understanding why it's not right is to realise that there isn't just a single on-off switch for cell division. If there was, then if any one of these mutations which in genes which are important in regulation, if any one of those mutations could lead to cancer, then we would have a huge problem, because as we've seen, there would just be too many of these mutations arising. So the way the body protects itself against that
is that there's a whole system of checks which is constantly trying to make sure that cells don't divide inappropriately. So there isn't just a single signal that sells to a cell to say, right, divide now. The cell has to get a whole series of signals, and it has to have a whole series of checks to make sure it's ready and able to divide, and that it really, really has had the signal to divide before it will finally do it. And the consequence of that is that a single mutation is never going to be enough to turn a cell into a cancer cell. It needs to accumulate several mutations, the combined effect of which is to wipe out the regulatory mechanism, or at least massively impair it. So what we need to do is see how big an effect on this rather scary number we've got here we get once we take into account the fact that you have to have more than one mutation in the same cell. That's the important thing. Okay, there's no point having one of the mutations in one cell and a different one in a different cell. That's not going to contribute to the development of cancer cells. You have to have more than one mutation in the same cell. And let's just see how big an effect that has on our number crunching. So let's do another back of the envelope, incredibly crude calculation just to sort of get a feeling for this. So here we go then. Remember that in the liver, we reckon that on average a cell will have undergone maybe 45 divisions after 50 years. And that every time a replication happens, the probability of an error arising which affects the function of a protein, we estimated to be 5 times 10 to the minus 4. And now we've said, yeah, but most of those proteins are not relevant to the possible development of cancer, so the probability that this protein that is affected will have an influence potentially on controlling cell division also happens to be 5 times 10 to the minus 4. So the overall probability of there being an error that might have an impact on controlling cell division is the product of 5 times 10 to the minus 4 times 5 times 10 to the minus 4, which is 2.5 times 10 to the minus 7 for each cell division. Now, to work out the probability that we get 2 such errors in a single cell when we have 45 cell divisions, we can use something called the binomial distribution, which we're not going to go into here, but if you do statistics, is something that you will learn about, and it enables you to calculate the probabilities of combined events like this. And for a very small probability, it simplifies to this expression here, and it churns out a number of 6 times 10 to the minus 11 for each cell. So this apparently tiny probability for a single cell that it might accumulate 2 such mutations. But of course there are a lot of cells in the liver, so when we multiply that by the number of cells in the liver, which is 1.5 billion, we end up with 9 cells as our estimate, the number of cells in the liver which have actually got 2 such mutations, which isn't very many out of 1.5 times 10 to the 11 cells. But on the other hand, it's 9, and any one of those could potentially give rise to a cancer. So should we be very worried about that? Well, we've just arbitrarily said that's the probability of getting 2 such mutations, but 2 is not enough. If we come back to the picture that we had before, now remember that when we took the liver as an example, we estimated that the number of functional mutations, in other words, mutations that have potentially a significant effect on cell function in liver cells after 50 years, might be something like 3 billion. Now, of course, most of those won't have any effect on cell division because most of those genes that are affected have completely different roles, which in no way impact on triggering the division of cells or anything else linked to it. So we can sort of modify that number to take account of that. The total number of genes coding for proteins in the human genome is around 20,000. And from studies of the mutations that occur in cancer patients, we can estimate that in a typical cancer, there's probably only around 10 of those genes which are commonly mutated and which seem therefore to be important drivers of the development of a cancer. So if we just multiply our 3 times 10 to the 9 mutations by the fraction of genes which could be implicated in generating a cancer, we come up with a number of 1.5 million. So that looks like a very scary number because, again, that suggests there are 1.5 million cells there in the liver which seem to be poised about to lead to the development of a tumour. And obviously that can't be right. And the key to understanding why it's not right is to realise that there isn't just a single on-off switch for cell division. If there was, then if any one of these mutations which in genes which are important in regulation, if any one of those mutations could lead to cancer, then we would have a huge problem because, as we've seen, there would just be too many of these mutations arising. So the way the body protects itself against that is that there's a whole system of checks which is constantly trying to make sure that cells don't divide inappropriately. So there isn't just a single signal that sells to a cell to say, right, divide now. The cell has to get a whole series of signals, and it has to have a whole series of checks to make sure it's ready and able to divide, and that it really, really has had the signal to divide before it will finally do it. And the consequence of that is that a single mutation is never going to be enough to turn a cell into a cancer cell. It needs to accumulate several mutations, the combined effect of which is to wipe out the regulatory mechanism, or at least massively impair it. So what we need to do is see how big an effect on this rather scary number we've got here we get once we take into account the fact that you have to have more than one mutation in the same cell. That's the important thing. Okay, there's no point having one of the mutations in one cell and a different one in a different cell. That's not going to contribute to the development of cancer cells. You have to have more than one mutation in the same cell. And let's just see how big an effect that has on our number crunching. Now, this is not the development of a liver tumour. This is actually for colon cancer, but the principle is the same, that you can see that after you've had typically 2 mutations, you get a state where you have some abnormality and you get a cell which is occasionally managing to break free of the restraints and divide when it shouldn't, so that you get a localized region of abnormal cells in the liver or wherever. But it's not enough for the division to get really out of hand at that point. You need to accumulate more mutations for that to become significant. And once you start requiring 3 or more mutations before you say, right, well, this is now really seriously at risk of developing into a tumour, then those numbers start to look a lot more promising. So if we now extend our binomial distribution estimate to say, well, let's have a requirement that you have at least 3 mutations which could drive the development of a tumour in the same cell, then we now get a probability of about 2 times 10 to the minus 16 of that happening. And even when you multiply that by the large number of cells in the liver, that now only comes to about 3 times 10 to the minus 5 as the probability, as the number of cells that we would estimate. In other words, there's only a probability of about 1 in 300,000 that a cell in the liver would have that set of mutations. So now you can start to see that it's not unreasonable to see why cancer might not happen all that commonly.
On the other hand, of course, we know that cancer isn't as uncommon as all that, that you see various numbers, but possibly 1 in 2, 1 in 3 people might develop some kind of cancer during their lifetime. So we maybe have to think, well, perhaps we're being over-optimistic here. And the reason for that is that errors resulting from replication problems are not the only source of sequence errors in the genome. And what we haven't considered at all is that all the time the DNA is just sitting there in between replications, DNA could potentially become damaged. Reactions could happen which could modify the individual bases in the DNA. And then when replication does happen, that could lead to sequence errors in the daughter cells. And those kind of errors can happen as a result of radiation. For example, if you're exposed to ultraviolet light, it can affect the skin. If you have higher energy radiation, it can affect cells within the body. And it can happen as a result of reactive chemical species, some of which are produced quite naturally in cells. So radicals based on oxygen are formed quite naturally as a sort of byproduct of respiration. And although cells do a very good job of mopping them up and trying to make the probability of them causing damage to DNA tiny, it can't reduce it to zero. And other chemicals which we just become exposed to in our food or elsewhere in the environment, tobacco smoke is a famous example of this. Those chemicals too can potentially get into cells and produce reactive species which can damage the DNA. So that has a big increasing factor on the probability, which means that the probability of a cancer developing is not frighteningly huge, but it's not negligible either. So if we now extend our binomial distribution estimate to say, well, let's have a requirement that you have at least 3 mutations which could drive the development of a tumour in the same cell, then we now get a probability of about 2 times 10 to the minus 16 of that happening. And even when you multiply that by the large number of cells in the liver, that now only comes to about 3 times 10 to the minus 5 as the probability, as the number of cells that we would estimate. In other words, there's only a probability of about 1 in 300,000 that a cell in the liver would have that set of mutations. So now you can start to see that it's not unreasonable to see why cancer might not happen all that commonly. On the other hand, of course, we know that cancer isn't as uncommon as all that, that you see various numbers, but possibly 1 in 2, 1 in 3 people might develop some kind of cancer during their lifetime. So we maybe have to think, well, perhaps we're being over-optimistic here. And the reason for that is that errors resulting from replication problems are not the only source of sequence errors in the genome. And what we haven't considered at all is that all the time the DNA is just sitting there in between replications, DNA could potentially become damaged. Reactions could happen which could modify the individual bases in the DNA. And then when replication does happen, that could lead to sequence errors in the daughter cells. And those kind of errors can happen as a result of radiation. For example, if you're exposed to ultraviolet light, it can affect the skin. If you
have higher energy radiation, it can affect cells within the body, and it can happen as a result of reactive chemical species, some of which are produced quite naturally in cells. So radicals based on oxygen are formed quite naturally as a sort of byproduct of respiration. And although cells do a very good job of mopping them up and trying to make the probability of them causing damage to DNA tiny, it can't reduce it to zero. And other chemicals which we just become exposed to in our food or elsewhere in the environment, tobacco smoke is a famous example of this, those chemicals too can potentially get into cells and produce reactive species which can damage the DNA. So that has a big increasing factor on the probability, which means that the probability of a cancer developing is not frighteningly huge, but it's not negligible either. If we look at the particular mutations which have been found to be important in this particular pathway for development, in this case of a colorectal tumour, we can sort of see the kinds of functions that they have which get disrupted as a result of mutations. So APC codes for a protein which is involved in sort of controlling the way that cells in the gut epithelium are stuck together to make a coherent tissue. And it plays an important role actually doing various things, but in particular in regulating how often a cell can divide. And it provides an important brake on that, that even when cells are being told to divide, APC will resist that unless there's a very strong signal telling the cell that, yeah, you really do have to divide now. And so one of the very early mutations is that brake gets taken away as a result of APC losing its function. Now, KRAS, which is the protein produced by this second gene here, that one is different because that protein is an active promoter of division. And what happens is that normally it will only become active in a cell when a signal from outside, like a growth factor or a hormone or something, binds to the cell to deliver a message from cells around it that says, yeah, now would be a good time to divide. And normally the KRAS protein just sits there not doing anything until it responds to that signal from outside. And what happens is it then becomes active just for a very short time and starts to initiate production of other proteins in the cell, which can be used to drive cell division. But it only becomes active for a short time so that the whole process will only be seen through and lead to cell division if it's repeatedly being stimulated. And that's important because otherwise, it might just get some random molecule happening to bind there and, that's just there by chance. And if that triggered cell division, it might happen at an inappropriate time. But if it depends on having to just keep being re-stimulated, that's only going to happen when there really is a strong hormonal or growth factor signal coming in there. And what happens when KRAS gets mutated is not that it loses its ability to drive cell division, quite the reverse. What it loses is this off switch that switches it off after a certain time, so that it only needs to get stimulated once and then it remains active the whole time. And so it will tend to start driving cell division at times that are completely inappropriate. And it can, drive up a very rapid rate of cell division as a result of that. SMAD2 and SMAD4, these next mutations here, again, are involved in signaling pathways. So they're involved in controlling the rate of cell division by basically transmitting signals that arrive at the cell surface to the nucleus. So again, loss of functionality of those, or modified functionality of those, can affect the rate of cell division. Now, P53, which is the fourth one here, that one is probably the single commonest protein to be implicated, or the gene coding for the protein is the single commonest one to be mutated in a whole range of cancers across the body. And P53 has many functions, but essentially it's like a coordinating protein. It receives signals, in particular from proteins involved in DNA repair. So we've seen that errors in the DNA sequence can arise just quite naturally, not just during replication, as a result of environmental factors. And there is a whole bunch of DNA repair enzymes in the cell whose job is to detect those errors and to try to correct them. And in effect, there is a sort of monitoring process, and when such a repair enzyme is unable to correct a mistake, what it does is it recruits P53 to bind to it, and the P53 binding basically then signals that there is a problem. And at that point, P53 will basically stop the cell from dividing any further until the error can be repaired. So when there is a problem with the genome, most of the time it gets picked up by these DNA repair enzymes, and either they manage to correct it, or if they don't, they sort of effectively tell P53, and P53 then puts the brakes on very firmly and stops the cell from dividing. Obviously, the problem comes then if P53 loses that ability either to interact with the mechanisms that are signaling to it, or to put the brakes on cell division. And so P53 has a very powerful role in controlling the cell cycle, and when it goes wrong, it can be a very serious problem. The repair enzymes themselves are also often ones which are coded by genes which get mutated in the development of cancers. So those two, obviously, if you lose the ability to correct correct errors in the DNA, then you become much more susceptible to the potential of errors in key genes happening and tumours developing. Okay, so we've opened a huge box here, and we're not really going to go any further with it because it's an enormous topic. But I just wanted to reach this point so that you could see one of the many reasons why it's so important that replication is as accurate as it possibly can be, and also that there are other mechanisms in the cell for keeping the integrity of the genome, because when that breaks down, although the probabilities in any individual cell of anything dire happening are very small, there are so many cells in the body that there is then a significant probability of a problem and something like a cancer developing. Okay, so we will stop there, but obviously there are lots of areas of interest that you could go on and read more about if you choose to do so.